Hello everyone. Today we are going to continue with core compulsory theory paper Clinical Immunology. In credit 1, Determinants of Microbial Pathogenicity, we are going to deal with invasion. In the previous video we talked about cell adhesion. The development of an infectious disease in an individual involves complex interactions between the microorganism and its host. The key events which occur during an infection include entry of the microorganism into the host tissue, adherence of the pathogen to the mucosal surface, bacterial colonization and invasion inside the host tissue, evasion of the immune response elicited by the host, and lastly, tissue injury or functional impairment caused by the bacteria. Entry into the host cells is a specialized strategy adapted by many pathogenic organisms for survival and multiplication. Pathogens actively gain entry into the host mucous membranes and epithelium after attachment to the mucosal cell surface. This is accompanied by synthesis or production of lytic substances which alter or modify the host tissue by attacking the ground substance present in the basement membrane of integuments, intestinal linings, and destroying the cell surface receptors, also degrading the carbohydrate or protein complexes which are present in between cell junctions and on cell surface, for example the glycocalyx. Crossing the epithelial barrier is an important step. Without crossing the epithelial barrier, the pathogen cannot bring about a successful invasion inside the host tissue. Defective epithelial barriers are a point of entry for pathogenic organisms to colonize the host tissue. Catheters are a very good example of how microorganisms can penetrate inside the epithelial barrier to bring about invasive infections. Breaking down the epithelial barrier is a common thing in establishing invasions. In addition to catheters, other defects such as burns and wounds can predispose a patient to infection. A rare example is anatomic defects, as it is not as common as catheters which penetrate the wound directly or burns. If a patient has recurrent infections in the same anatomic locations such as the tracheophageal fistula. In the tracheophageal fistula, the two tubes, that is trachea and the esophagus, are connected by a fistula. In this case, this fistula can carry content material from one tube to another. So here the oral secretions or content can be carried from the fistula not only to the stomach but also in lungs. Breakdown of epithelial barrier can be brought about by extensive use of drugs or chemotherapeutic agents to treat cancer. An example is mucositis caused due to extensive chemotherapy in the gastrointestinal tract. This predisposes the patient to various systemic infections which can either be caused due to microorganisms which invade from outside or opportunistic infections due to the own microflora. Some microorganisms can cross the intact epithelial cells to establish an invasive infection. One mechanism is to invade and enter the specialized cells of immune system which carry out a job of transiently opening outside the epithelial cell junctions to sample and survey the mucosa for antigens and viruses. In the diagram, you can see a dendritic cell in brown color. It is basically trying to sample the mucosa by extending processes between the intercellular junctions of the epithelium. A very common example is viruses. So viruses cannot enter the cells directly as they lack the receptors, but they invade the dendritic cells so that they can be brought across the epithelial cells. From here, the viruses can spread to other cells. Another specialized cells which can survey the epithelium and sample the content of the intestines are M cells. Such M cells are present on surface of Peyer's patches of the intestine. Pathogens like Salmonella invade the epithelium through M cells and cross the intact barrier. Bacterial pathogens can enter the surface of the epithelial cells by more unreceptive or passive mechanisms which are not directly related to the pathogen itself. They can enter into the host tissue using small breaks, lesions, ulcers, or wounds in mucous membrane. They can also enter into the body of the host using arthropod vectors which feed on the host blood. Another specialized example is that of active invasion. Certain parasites like Plasmodium toxoplasmosis use a mechanism of active invasion in which they squeeze themselves into the host cells. Many viral infections are caused due to entry of virus by receptor-mediated endocytosis. After entry into the host cell, they then invade the cytosol. Another specialized mechanism is invasion of the Mycobacterium tuberculosis into the macrophages with the help of phagocyte. Once inside the phagosome, the tuberculosis bacteria can survive there for a long period of time and can evade the host immune response as well. Once under the mucous membrane, the bacterial pathogen may penetrate into deeper tissue and continue to spread, disseminating throughout the body of the host. One way the pathogen does this is by producing specific products or enzymes to promote its own spreading. These products can be virulent factors or enzymes. Bacteria can also enter small terminal lymphatic capillaries that surround the epithelial cells. These capillaries then join or merge into the lymphatic vessels which eventually enter the circulatory system. Once the circulatory system is reached, the bacteria have an access to all the organs in the host tissue. Okay. Now we are going to talk specifically about invasions. The invasion of host by pathogenic organisms may be assisted by synthesis of bacterial extracellular substances. These extracellular substances are known as invasins. Invasins act against the host by destroying the primary and the secondary defenses of the body. Usually, invasins are enzymes, that is, proteinaceous in nature, and they act on a short range, that is, in the immediate surrounding of bacterial growth, and may not actually kill the cells as a part of their range activity, but they facilitate the growing and spreading of pathogen. The damage to the host as a result of invasion activity may actually become the part of pathology of an infectious disease. The extracellular proteins produced by bacteria which promote their invasion may not be clearly distinguished from extracellular protein toxins or exotoxins which can also bring about damage to the host. Invasins are short range and may not actually kill cells, but exotoxins are cytotoxic and can act on distant sites from where they are secreted. Also, the exotoxins secreted are very specific and potent in their activity. Spreading factors. Bacterial enzymes which have the capability to bring about changes in physical property of tissue matrices are called as spreading factors. One of the original spreading factors produced by Streptococci, Staphylococci and Clostridia is hyaluronase. It attacks the intestinal cement and the connective tissue by depolymerizing the hyaluronic acid. Another spreading factor synthesized by Clostridium histolyticum and Clostridium perfringens is collagenase. Collagenase breaks down collagen. Collagen forms the main framework of the muscles. Breakdown of collagen facilitates gas gangrene in the organisms affected by Clostridium. Yet another spreading factor synthesized by Vibrio cholerae and Shigella dysentery is neuraminidase. Neuraminidase depolymerizes neuramic acid, also known as sialic acid, which is an intercellular cement of epithelial cells of the intestinal mucosa. Yet another enzyme produced by Streptococci or Staphylococci is streptokinase or staphylokinase respectively. They bring about conversion of inactive plasminogen to plasmin. As you can see in the diagram, plasminogen in the inactive form is converted into active plasmin. If plasmin is present, the fibrin protein, which is an essential factor for clotting, converts into fibrin split proteins. The relative absence of fibrin in spreading bacterial lesions allows more rapid diffusion of infectious bacteria. Staphylococcal coagulases are synthesized by Staphylococcus aureus. They can be present in either a cell-associated form or a diffusible form. Coagulase activity is usually related with pathogenic species Staphylococcus aureus, whereas non-pathogenic species of Staphylococcus do not have the cell-bound coagulase activity. It is made possible that the cell-bound form could offer an antigenic disguise, or a clotted fibrin on the cell surface, or a streptococcal lesion encased in fibrin such as pimples or boil can cause bacterial cells to be resistant to phagocytosis or tissue bacteriocytes or even drugs which might be unable to diffuse to their bacterial targets. Enzymes which cause leucolysis or hemolysis. A number of enzymes which help in formation of pores in the cell membrane or which attack the phospholipids present in the cell membrane come under the category. They bring about hemolysis. Hemolysin is a spreading factor synthesized by Staphylococci, Streptococci and a number of species of Clostridia. Their main function is to rise the red blood cells. In this category, another spreading factor which is synthesized is phospholipase. Phospholipase is produced by Clostridium perfringens and it hydrolyzes the phospholipids in the cell membrane by removing the polar head groups in the cell membrane. Lecithinases are synthesized by Clostridium perfringens, also destroy lecithin, that is phosphatidylcholine, which is present in the cell membrane. Leucocidins are synthesized by Staphylococci. Leucocidins basically disrupt the neutrophil membrane and cause them to discharge the lysosomal granules into the cell. Streptolysins, synthesized by streptococci, repel phagocytes and disrupt the phagocytic membrane, again causing them to discharge the lysosomal granules into the cytoplasm. Both these enzymes are considered to be bacterial exotoxins. Another class of spreading factors is the extracellular digestive enzymes. These enzymes are secreted by heterotrophic bacteria. These heterotrophic bacteria synthesize a number of enzymes such as proteases, lipases, glucohydrolases, and nucleases. They do not have a direct role in invasion or pathogenesis, but these enzymes help in other functions related to bacterial nutrition or metabolism and can also aid in invasion either directly or indirectly. Toxins with short range effects related to invasion. Some toxins are known to have adenylate cyclase activity and can have immediate effect on host cells that support bacterial invasion. A component of anthrax toxin known as the edema factor is the adenylate cyclase. Adenylate cyclase causes an increased level in the cyclic AMP and disrupts the osmolarity, thus rendering the cell permeability and increases water retention, that is edema. Toxin of Bordetella pertussis, which is the agent of whooping cough, has a similar effect. As you can see in the diagram, the bacillus releases the three components of the exotoxin: the protective antigen as shown in brown color, the edema factor as shown in blue color, and the lethal factor as shown in green color. The PA component binds to the ATR receptor. With the help of furin, a part of the PA component is clipped off. After that, the receptor and the PA component form a PA heptamer. To this PA heptamer, either the lethal factor or edema factor can get attached as shown in the diagram. The next step is endocytosis of this receptor bound edema factor or PA factor. Edema is seen as a pathology because it increases the CAMP in the affected cells and disturbs the cell permeability. As you can see, once the edema factor is released, it activates adenyl cyclase present in the cell membrane, which converts the reserves of ATP inside the cell and converts it into CAMP. Once CAMP is increased, it increases the water retention capability inside the cells, leading to edema. If the lethal factor is released, it causes degradation of the mitogen associated protein kinase, that is MAP kinase, which ultimately leads to cell death. Process of microbial invasion. Traits which increase an invader's chance of success are known to be high growth rate, a good dispersal capability, phenotype plasticity, and genetic diversity. For a successful invasion, which is depicted in a 4-step process, can be envisioned from the perspective of both the invader and the resident. The triangle microbe represents an invader that is introduced into the community with various microbial taxa depicted in different colors. The same community is represented as it transitions through each phase of invasion process. Each phase is a key step with a particular barrier value, meaning that there may be a large or a small barrier in path of successful invasion. From an invader's perspective, Many listed factors may aid in bypassing the barriers of invasion resistance. Should an invader be able to withstand the abiotic pressures of new environment, it is considered to be introduced. Biotic resistance will keep the invader in establishment phase as it maintains a viable population. If the invader gains access to resources that will support its growth, it will pass to the growth and spread phase. Growth and spread of invader may cause impacts to the resident community. That is, after a successful invasion, displacement or shift in the resident taxa may be seen, and also the functioning of the community may be altered. Enteroinvasion. The 4 genera of Enterobacteriaceae are known to invade and penetrate inside the intestinal cells and can cause disease. These include Shigella, Salmonella, Yersinia, and E. coli strains. Shigella strains are strictly enteroinvasive. They enter and kill the intestinal epithelial cells. Shigella are known to cause localized ulcerations, bleeding, elicit an inflammatory response that results in watery stool that contain pus, blood, which is characteristic of dysentery. Some Escherichia coli strains also have classical enteroinvasive O serotype, which use a similar pathogenic mechanism as that of Shigella. Salmonella species invade the intestinal epithelium. Salmonella is known to cause localized inflammatory lesions that result in profuse diarrhea. Salmonella chloriosus and Salmonella typhi survive and proliferate within vacuoles in the epithelial cells, move to lamina propria, and hence enter the lymph node and possibly the bloodstream. The severity of disease caused by Salmonella typhi is worsened by the ability to survive within the macrophage by its phagocytic mechanism. Yersinia pseudotuberculosis and Yersinia enterocolitica are also enteroinvasive. The symptoms include diarrhea with blood abscesses in liver and spleen, appendicitis, and mesenteric adenitis. Yersinia pertussis illustrate another form of invasion where extracellular intestinal infection is limited by chance rather than active penetration of gut epithelium. These are the references.